Okay, hello everybody. My name is Camilla. I am a graduate student in physical oceanography at UH Manoa. I'm getting my master's, and my favorite place is, I would say, the beach. And I'm really interested in how waves and wave energy interact with the coastline. So today I will be talking to you about how some surf can hurt the beach, but how forecasts can help us prepare. The motivation for this study are the current impacts, specifically in West Maui. In the middle, you have a map of Maui, and here our study focuses on the west portion of Maui, where the impacts of wave erosion are very evident. On the right, on the bottom left, right here, you can see a picture of the tree line falling into the ocean after a big wave event. And here on the left, you can see a stretch of coastline where there is no more beach and there's no more sand, so the waves are digging straight into the dirt, which is spreading around the reef, which is very bad for the reefs. And here on the top left, you can see where the waves have undermined under the coast, and this is a lanai of a building that is collapsing. And there's lots of hotels that are, getting brown salt water into their basement. And it's also a public safety issue because, as we can see here on the right-hand side, the waves are washing up right to people's homes, and this little hale right here, which was a pool hale, has been demolished since this picture was taken in June because it became a hazard. Up here, we have a picture of the main road in and out of West Maui, and we have waves crashing right onto the road. And here on the bottom, we can see the monk seal is on the road because there's no more beach for her to go. So these impacts in West Maui are giving us a look into the future of climate change, where sea level rise will be affecting more and more coastlines. And in Maui, we're already seeing some impacts. So my research project focuses on wave runup. As you can see right there, this wave is washing right onto the highway in West Maui. And so what is wave runup? Wave runup is the maximum height the water reaches up the shore. So this is a picture of the north shore of Oahu. We don't have too many pictures of West Maui yet, so I'll be showing some examples from other places. So this is a wave running up onto the shore and overtopping on this unfortunate home. So there are many things that influence wave runup. Here we have a diagram. We have sea level, and then we have the shore. And here is indicated the run-up height, or where the water reaches onto the beach. So of the many things that contribute to run-up, we have our swell, or regular waves, which can break offshore at the reef, or they'll break straight onto the beach, depending on the bottom. In Hawaii, we mostly have fringing reefs, especially on Maui. So after the waves break around the reef, so actually we can predict waves. We can predict swells with wave models. This is off of a website. And what this is showing is the color scale is feet in wave height. And it goes from, the blue is 1 foot to 30 foot in the purple. And the arrows show direction. So you can see that around West Maui, there's some complicated interactions with the waves. And that's why it's a very interesting place to study. And so the swells is something that we can predict. There are other factors caused by the swell. So for example, as the waves break, they don't just disappear. There's some energy that is transferred into the shore that we call wave surge. And there's these smaller size waves, but they have longer periods, which I will explain. I am going to show you a video in which I will show you a surging wave. It's going to play for about 8 minutes. So what you will see is in the background, you'll see the regular swells. And then 2 times during this video, at minute 220 and at minute 740, we will see this surge wave. So watch out for that. So here we just have our regular waves. And so they break, every 10 to 20 seconds, you have a normal swell, and then there's a swell surge. And so these surges come every few minutes. So this is an 8-minute video, and we have 2 waves coming in. So it's going to come up in a couple minutes too. Another surge wave right there. So this is a phenomenon that's hard to see, but it's there. And if you watch carefully in certain bays and areas, you'll see these long period waves. So another thing that the swell does after it breaks is it pushes all these waves coming in, push all the water towards the shore and actually elevate the sea level towards the shore. And this we call setup. So these are all phenomena caused by swell. There are other factors contributing to the wave run-up as sea level variability. And so we have the tides. And of course, tides are variable. We have low tides, high tides, and we have times in the month where the high tides are higher and the low tides are lower, and these depend on the moon phases. And on top of the tides, we also have sea level variability. And this encompasses a whole range of phenomena. And this is a long-term sea level trend of the Kahului tide gauge in Maui. And here we can see these are monthly averages of the water levels. And so we see the red is an overall increasing trend of sea level rise, which will be continuing into the future with climate change. And then these variations, these ups and downs in sea level are caused by interannual variability of seasonal changes such as El Nino patterns and things like that. So all of these components added together contribute to our run-up height, which can get pretty far upshore if the conditions are right. If you have a big swell, a high tide, a high sea level, you can get some pretty big run-up, even if you don't have a storm or anything like that. So for our project, thanks to the Sea Grant, we'll be collecting field observations to help us calculate wave run-up. So we'll be measuring waves along West Maui. This is a map of West Maui with all the locations where we are currently have instruments deployed along the shore. So the instruments we're deploying, sorry for the small print, I will guide you through this figure. First of all, we have these water level sensors. These measure the pressure of the seawater, and from this we can calculate the waves. We also have these current meters, which measure the speed of the water moving by emitting sound waves, by acoustic. And then to secure these instruments into the sand, we're using this sand anchor tube in which we've put the little yellow sensors, and we're holding it down with these fiberglass grates. This is an image of one of our instrument deployments on West Maui. These are our UH divers, and right there you can see the little sand anchor tube with the grate on top with the pressure sensors in it. We did about 25 of these deployments, some by scuba and a lot by snorkeling. And now we'll be going back in a few months to collect the data. So we're collecting all of these data because one of the people on our research team has developed a wave run-up model specific to West Maui. And this is because West Maui has a very particular bottom geography of the bottom ocean floor where there's a lot of ridges. And so these images are some of these model outputs that show how the red colors are high energy and the blue colors are low energy. So it shows how when the waves arrive close to West Maui, all these ridges at the bottom of the ocean floor cause the waves to bend and the energy to arrive at certain places onto the shore. This one is for a south swell, and this one is for a north swell. And the data we're collecting is going to help us validate and make the model work better. And we will be using that to help us prepare for days like this. This is an image taken in Waikiki, and the wave is crashing right into a lanai that is not meant to be a breakwater or anything like that. Where these people are standing is where the beach usually is. And as you can see, this is a sunny day. There's no storms, and yet we have this really impactful event. So to prepare for these days, this is what a wave run-up forecast looks like. This is what we're developing for West Maui. This is what is currently in use for Waikiki. We have these for a few locations on this PAC-IOS website, which we're collaborating with this organization. And so this forecast, this graph on the left shows, so the solid line is the expected tide level, and the dotted line is the tide plus all of those other components that contribute to wave run-up. And so here we can see that the forecast shows that there will be, you see the red line shows the threshold for beach flooding. And so we see there's a few days where the waves will be flooding the beach. And to validate these forecasts, we've partnered up with the Hawaii Sea Grant King's Tides Project, in which citizen scientists are taking pictures of these high water level events. And this photo was taken exactly at the time where this red arrow is, therefore validating the forecast and showing that, yes, there was beach flooding on that day. And that's the very important aspect of our project, is involving the community to from Maui to take pictures and to help us validate our data, because as we go, we're going to learn more and more about these processes. And so the goal of this study is to increase community resilience by empowering community managers with the knowledge of when these things will happen, because they have to put in sandbags, they have to prepare. There's one road in, one road out of West Maui. It's a public safety hazard. And so the best we can do right now is prepare and understand how these phenomena will be affecting us into the future. And with that, I'd like to say thank you to the Sea Grant and all the people involved in this project. Thank you.
Okay, hello everybody. I'm Claire Lewis. You might notice I have an accent. It's because I'm originally from the UK. I came to Hawaii to pursue my PhD at UH Manoa, where I'm looking at tropical coral reefs. And there aren't very many of those in the UK, so I thought it would be better to come here. Before we get going, I just want to acknowledge some really important people in this research. Sea Grant, who funded this whole thing and pay my salary, so they're very important, as well as the Hawaii Division of Aquatic Resources and Nui Nui Coral Restoration Facility. The staff at that facility were essential to getting this work done. So before we get going and thinking about corals and coral restoration, I want to just draw your attention to a decision-making framework, which we all use all the time, every day, but you probably don't think about it, which is that when you're facing some stress or some discomfort, you're faced with three possible outcomes. You can either move, leave, go away, up sticks, and get away from that stress. You can make some kind of change, adapt to remedy your current situation, or you can just sit there and suffer and kind of deal with it. Obviously, that last one's the least preferred. And we use this all the time. So for example, let's say this weekend is a really nice sunny day and you want to go to the beach, perhaps Waimanalo Beach. But it's midday and it's actually really sunny, perhaps too sunny, and you're uncomfortable. You're squinting, you maybe feel like you're getting burnt. So what do? Well, you could potentially move, perhaps to this nice shady portion of the beach over here, where it's more comfortable and you'll be out of that excess light. You could make a change. Perhaps you brought your hat or your sunglasses with you that day, or maybe even an umbrella, and you're going to make your own shade to make you more comfortable. Or you could just sit there, tough it out, maybe go home with a headache at the end of the day, or even perhaps, if you're very pale like me, get burnt. Now, for me, that's probably about the worst thing that's going to happen if I'm in a scenario where there's too much light. But for these animals that live off the water here in these coral reefs, excess light can be really damaging and potentially life-threatening. So we've got this beautiful close-up of a coral here, and you can see its tentacles. And if we zoom in on those tentacles, you can see that it has these little greeny-brown circles all over it. And these are photosynthetic algae that live inside the coral tissues and make food and sugar for them. Now, normally, this is a very successful partnership. When conditions are right, this enables corals to proliferate and create huge structures like the Great Barrier Reef. But this is a relatively fragile relationship. And if the conditions become stressful, and that can be too much light, too much heat, pollution, any of these conditions, and it's important to know that that's different for different species and different individuals of corals even, that can throw this relationship into disarray. And instead of producing those food and sugar, these algae produce toxins. And so the corals expel them, get rid of these algae, and so instead of them being this nice brown color, you're left with clear tissue over the top of white skeleton. And so these corals end up looking bleached as a result. And effectively, when these corals are in this state, they're starving. They're not getting any food. So they can only be in this condition for a short amount of time before they die, effectively. So this is a really bad scenario. So I think it's safe to say that we could consider excess light a stressful situation for some corals. And so they have that same 3 potential scenario outcome. But that first one isn't really an option. Corals are rooted in place. They are literally making rock as they go. They can't just move to somewhere better. So instead, they're left with adapt. And that's really what I'm going to be talking about today, acclimating to different conditions, or the potentially dire situation of just sticking it out and dying. So what I'm really going to be talking about is how corals have come up with some really ingenious ways of acclimating and handling excess light. And I'm going to do that by talking about this really wonderful coral, Pavonovarians. Now, these are found in reefs all over Hawaii. They're not, the flashiest of coral. Normally it's this one. I know you were thinking it's this one. It's not. It's these kind of unassuming encrusters that live on the rock here. But when you get up close, they're really quite beautiful. And the defining characteristic of these species are these seemingly random ridges that form all over the skeleton. And these ridges can be straight, they can be curly, they can be curved, they can be tall, they can be low, and different corals can have really different appearances, and that's really where this species gets its name. Its variable appearance makes it varians. The other interesting thing about this species is that it can live in a really wide variety of light environments, extremely wide. You can find it in just a few inches of water down to 200 feet. And it can be found in full open sunlight like this, as well as under ridges and in crevices and caves. Now, what is it that makes this coral able to withstand such an extreme range of light environments? I think the secret is in these ridges. And I'm going to kind of explain that point using this individual here, which I collected from Kaneohe Bay. It's doing pretty normal Pavona stuff, just encrusting over a ledge. And when I took off this end portion here, which is this kind of point here, you can see that the skeleton is really complex. There's a lot of 3D structure. It's almost all ridges and valleys. There's no real flat portion to the skeleton. But when you look at the underside of the exact same individual of coral, it looks completely different. And you can see that probably the most obvious and noticeable thing is it's a lot darker, and that's because there's a higher density of those photosynthetic algae. But when you look at it a little bit closer, you'll also notice that those ridges are far less pronounced. They're much smaller, they're thinner, and we now have areas of just flat skeleton in between those ridges. And so I thought this was a pretty clear smoking gun that these corals are responding to light and changing their morphology in a potentially adaptive way. Now, me just kind of taking pictures and thinking that's probably what's happening is not enough for science, so I had to do some experiments. Now, the really nice thing about corals is that you can break them up into little pieces and they'll keep growing. So you don't have to worry about genetic effects. So I looked at 4 different individual corals. So this is one coral, 2, 3, 4. And each of these fragments is genetically identical to the one next to it. So when we run this through an experiment, we're removing any individual effects and just looking at the environment, the light on how it changes. So I had these 4 individuals. I split them up into these different fragments, which I tried to make sure were equally sized as well as equally ridgy to start with. And I put them in 3 different light environments. This first one was just full sunlight, and you can see that's one of those tiles there with the coral growing on it. So full sunlight. I then did 2 layers of 50% shade cloth, which would be this one, and 4 layers of 50% shade cloth. And as a gauge, these corals are experiencing about 1/10 of the light as those in the full sunlight. So it's a really big environmental difference that they're experiencing. I ran this experiment for about 6 months, and I recorded a host of growth metrics as I was going through this. And I really wanted to get at how do they grow differently in high light versus low light, and particularly understand this ridgy component and if they're changing that. Well, the results were pretty striking. So I'm just going to talk you through what we are looking at here. So these corals in the top were all grown in full sunlight. These in the bottom were all grown under 4 layers of shade cloth. So that's sunlight and shade. And this is individual coral 1, 2, 3, and 4. So this coral is genetically identical to this coral. The only thing that's different here is that it was grown under those different light conditions. But as you can see, this is a pretty staggering difference in growth rate for the only thing to be different is light. And actually we find that this species goes 3 times faster in the shade than it does in the sunlight. And this is a pretty big difference, and it's also pretty unusual for corals. Generally, they grow slower as light gets darker. So this in and of itself is somewhat unusual. But what I really wanted to get at was this ridges situation, right? Need to know how ridgy are they compared to another. And that's a really difficult thing to measure. You can't just get a ruler out, you can't even use calipers. So I had to get a little bit creative. And I did it using this technique called photogrammetry. And this is where you take multiple photographs of the thing that you're trying to make a model of. So this is about 40 photographs, and then you put it through some very fancy and expensive software, and it aligns all of those different photographs relative to one another. So this is a still, this is a still, these are all different photographs. The program then is very clever and basically creates an ever more dense and complete mesh of the object of interest. And the whole thing takes about 4 to 6 hours. By the time it's done, you end up with photo models that look like this. So this one here is the photo model. This one is the actual photograph. And then you can see that it's getting most of those features there. But the thing that's really great is you could kind of flip this on its side and measure the height of the coral if you wanted to. You're getting it in 3D. And what this enabled me to do was to take the surface area of the 3D fragment and then the surface area of the 2D fragment and compare the two. And if it has the higher the difference, the more 3D, the more complex that coral is. And what I found was that they are 2.5 times more 3D, more ridged in that full sunlight environment than they are in the shade. So what I predicted, that idea of having more ridges, higher ridges, did seem to come through in the data. And hopefully you can see that in these 2 different images. We can see that these ridges are very tall and very pronounced, whereas here on that same initial starting piece, they're really narrow and they haven't put a lot of skeletal growth into that portion. So this was really exciting. And that made me come back to this original kind of framework that we had, where they're not moving and they're not suffering, but are they really adapting? Well, they're certainly growing slower in the sun, which is a surprise, but they're also growing differently in the sun. They're growing more and taller ridges, and they're putting more of their skeletal mass into growing these ridges. And we hypothesized that this was because this species is self-shading. They clearly prefer the shade. They are growing faster and better in the shade. And so to tolerate these high light environments, they are creating their own shade via these ridges. And you can think of this as somewhat like a sundial. The more ridges you have, the more shade you generate across the skeleton. So as the sun moves across the sky in the day, the more ridges you have, the more portions of the skeleton are in shade throughout the day, and it makes it more tolerable for this species. Now, I think this is just pretty cool and interesting, but it's also useful. So as I said at the beginning of this talk, we worked closely with the Hawaii Division of Aquatic Resources and Nui Nui Coral Restoration Facility. And at this facility, they grow corals as quickly as possible under perfect conditions in tanks to outplant them onto reefs. So they are trying to restore damaged reefs and replenish them by using farmed corals. Now, what they really need to know is what the optimal conditions are for rapid growth. They are trying to grow these corals as quickly as possible. And what's interesting about this species is that This is a very different condition to a lot of other corals. So it's useful for these managers to know how best to grow these corals for them to grow quickly. But not only for them to grow quickly, but how to get them to survive best once they're out in the field. It's no good growing lots of very flat colonies of this coral because they'll just get into a high light environment in the wild and perish because they're not adapted to that environment. So now they know that just growing quickly isn't enough. They then need to be introduced to an appropriate light environment for where you know they're being outplanted. And that enables them to grow the ridges so that they can then survive and encourage that healthy reef system to grow into the future. So they could look at light conditions in very different parts of Hawaii, say Waimanalo versus Kaneohe Bay versus the outer reef, and get those light conditions and then grow those corals so that they are appropriately ridged for those outplanting conditions so that they can have the best chance of survival. And that's important because we all care about Hawaii's reefs. And as we enter a time of changing climate, we need to make sure that we can keep reefs healthy and resilient moving forwards as the climate changes and it becomes more difficult for them to do so on their own.
Hi, my name is Mykle Hoban. I'm a PhD candidate at the Hawaii Institute of Marine Biology, and that actually provides a pretty good segue for me. So why do we monitor coral reefs? The idea is we want to sort of keep this sort of thing from becoming this sort of thing, which is increasingly a problem. And this is in aid of management. So there's all sorts of different goals of management, but in this case, this quote sort of describes the goals, the management goals that we're trying to aid. Essentially, maintain marine ecosystems in a healthy and productive and resilient condition. And the key word here is that it sort of satisfies some desire that we have about that, the state of that ecosystem, but mainly it's healthy, productive, and resilient. So management relies upon monitoring, which essentially means counting things, right? Getting in the water and counting what is there. Because if you don't know what is there and how it's changing, you can't make decisions about how to interact with it. So in practice, what this often means is counting fish and counting corals, because these are the things that are most easily accessible and you can look around and see them. But the practices that we currently have in terms of monitoring have some drawbacks, namely that it takes a lot of time in terms of training. You have to know what you're looking for in order to count it. There's a lot of species to memorize. It costs money. You have to pay those people. You have to, invest in scuba gear and boats and all those sorts of things. And it takes time. This sort of wraps up with those other two. And finally, reefs are more than just fish and corals, right? So this is a small example of the many things that live out there that you're not counting when you're sticking your face in the water and counting things. Which brings me to this tool that we're trying to use. So environmental DNA is the idea that, so using this little cartoon here, it's the idea that if you look at this shark swimming around in the aquatic environment, bits of it will slough off into the water. And this is true for whatever lives in the water. So it could be skin cells or mucus or fecal matter or eggs and sperm or whatever. And the idea is that we can collect the water and we can attempt to pull the DNA out of it. And by looking at the DNA that was floating around in the water, we can attempt to reconstruct that community. We can see what has been swimming around in there and what it has been sloughing off of. So this is sort of a little overview of our kind of workflow with this. So I'll take you through this. So the idea is that we collect some water. We filter it through a very fine mesh filter. We extract and purify the DNA. And then what we try to do is we look at specific parts of that DNA that we think are good at identifying what species they are. We put them all together into a sequencer, which gives us a massive bundle of sequences back of all of the different stuff that's in there, which we then separate back according to what we started with. And then the key here is we try to assign those sequences to the species that they represent. So this is a pretty critical step. And so the idea is that we need some kind of, and so we call these sequences DNA barcodes because a specific sequence represents a specific species, like a barcode on a shampoo bottle. So the idea here is that we need some kind of database that tells us, okay, which this sequence goes with this species. And there's a number of options that currently exist that we can use. So these are some examples. There's government-run NCBI. And then there's more local ones. So Moorea Biocode was one developed for Moorea in French Polynesia. The Smithsonian and HIMB worked for one in Kaneohe Bay, and we're working on one for Hawaiian fishes. So we're still in progress with this. So let me lead you through sort of what we're doing. So one of our first main questions is, can we detect using eDNA, environmental DNA, can we detect the effect from existing marine protected areas? So those are areas that, like Hanauma Bay, where fishing and stuff is restricted. So these are sites around Oahu that are restricted in some way or another. So Pupukea, Waikiki, Hanauma, and HIMB. And then a reference site next door that's not protected. And then we're also looking at a couple of sites on the other islands, and we're attempting to look at the eDNA and see if we can detect an effect between the protected and the non-protected sites in terms of what is basically swimming around in there. So are there more of one kind of fish versus another? Secondly, we want to see, how does this compare to visual surveys? So this is something that's been going, it's integral to management. It's been going on for a long time. We have a lot of records of people sticking their heads in the water and counting fish and corals. So how does the environmental DNA, the communities that we construct from that, how does that relate to what people are seeing? Is it ultimately a useful and viable tool? And finally, can we look for things that are rare, endangered, or potentially invasive? Things that might be hard to see on any given moment, but might leave remnants of themselves that we can pick up. So I have here the hapu'upu, the grouper, the spiny lobster, which you don't see a lot of around here, and then our Roy, the invasive from Tahiti, just as examples of the sorts of things that we are trying to maybe, pick up. So ultimately, what we hope to get out of this is we hope to develop a tool that could be potentially rapid, potentially cost-saving tool for monitoring biodiversity, which we hope to complement, not necessarily replace, but complement existing tool sets, just to put another arrow in the quiver of ways that you can sort of keep a handle on what's going on in the ecosystems that you manage. We'd like to hopefully maybe be able to detect species invasions. So these are things that come from shipping or pet releases and all sorts of things like that. If we see this thing show up in the DNA early before it starts going everywhere, perhaps there's some remediation we might be able to do. Enable early detection of biodiversity loss, and not just loss, but change in terms of what's happening with changing conditions. So coastal development, runoff, all these sorts of things, that climate change, all these sorts of things potentially have an effect on the marine biodiversity. And can our tool help us get a handle on that? And then finally, we're working on developing a database of local marine species and their DNA barcodes. So a lot of them are already in databases, but there's a lot of things that exist only in Hawaii. I think there's a poster over there about endemics, and those are not super well represented in these databases. And so developing this will generally be quite useful for this sort of work, as well as just generally identifying things. And there's some collaborations here, as I said, with the Smithsonian and HIMB, trying to get a handle on what are the species that live in Hawaii and what are their characteristic DNA sequences. So that's where we are with the project. I just wanted to thank collaborators and Sea Grant, especially as a funding source. And I think I can ask for questions since I'm the last speaker.
Okay, let's have our speakers up in the front. And let's open up for questions. Does anyone have any questions? Yes.
eDNA. How does it reflect biomass?
Probably not very well. What you can potentially get from it, though, is relative abundance, how much of one thing versus another. So it's something that people are still really trying to work out. But ultimately, that will be one of its major sort of shortcomings, is that it's unlikely that it will give you a true sort of sense of what the biomass is.
investigated pressure. The pressure measures.
Mhm.
The waves.
Are they respond instantaneously or do they adapt? And what do you think the corals do? Respond in any way instantaneously or
So we're measuring the pressure as in the height of the water above the bottom, and we're just getting from that, we're getting the wave heights. As far as the effects on corals on that, I'm sure there's some effect. I don't know anything about that though. I'm just studying the waves, but I think the corals, they do sustain all that pressure, especially when the waves get big. Does that answer your question?
Yes.
Okay.
Did you have a question?
Yeah.
So, you know how the acidity in the ocean is going up?
Mm-hmm.
Do you know about any corals that could like survive in the acidity?
Right, how they can keep going when the water is getting more acidic. So again, different corals, different species handle the acidity better than others. So some will be able to tolerate it to an extent, whereas others will die. But there'll be a point at which none of them can handle it. It's just whether or not we can do something about it before it gets to that point. Yeah, it's sad.
I have a question about king tides.
Yes.
I grew up here at the term, and we never ever heard the term of a king tide. You know, we were high tides and low tides. And so what? All of a sudden, it's very important. It's a big deal. How did that come about? Did we always have king tides and we just didn't know it?
So the king tide phenomena is a combination of a very high tide. I think we've always had naturally occurring higher tides at certain times of the year. But what is happening now is as the sea level is slowly rising, these high tide events are becoming a little bit higher, imposed on the sea level rise, as well as all these other seasonal patterns of ocean sea level variability. There are a lot of these patterns that are very complicated, but they are increasing the mean level of the ocean. And so, yes, we are going to see more and more of these events into the future. They will become a common occurrence rather than a once in a while, unfortunately.
Yes.
As well. So, what are some of the solutions in place in kind of obviously stop the waves from
Yes.
well, I mean, there's other barriers, I guess.
Yes, on that side, I think there's a lot of engineering solutions for that. There are also solutions in which you would allow the beach to have more space, such as, we have all our roads are built along the beaches, and these prevent the beach, the sand from moving. So as far as solutions, I'm not sure what we're gonna do, but we'll have to do something because it's coming, and that's a very good question. We'll have to figure it out for sure. Yeah.
Any other questions?
Yeah.
Go ahead, you can go.
Okay. Michael, for the DNA testing, is there like a timeline on that?
Yeah, it varies. So it varies depending on local conditions. Obviously, water movement can move it around and stuff like that, but it tends to last between 12 and 48 hours. So it goes away pretty quickly, which means that what you're getting tends to be a pretty good snapshot of what's there. That's the hope in most cases. And so it depends on how quickly that microbial community basically will degrade the DNA that's there. Yeah, I mean it's a energetic it's a good food basically for microbes. There's sugar and there's phosphate and there's all sorts of stuff that they love to eat. So, and it turns out that in waters like ours that are clearer, less nutrient rich, it goes away quicker. So, which means that you're getting a more recent snapshot when you collect it.
Yeah, nobody's asked that coral lady a question.
He asked her one.
Oh, okay. So are there different species of Favona corals, and would they have, some have more tolerance to sunlight?
Yes, and yes. So there are lots of different species of Pavona, and they have very widely varying responses to light. Pavona varians is the most widespread and the most able to tolerate extreme differences. So there are other species that you really find almost exclusively in deeper water. There are some you find almost exclusively in those shallow, lighter areas. Pavona varians is really the one that can handle all of them all of the time, everywhere. It's like a real jack of all trades species.
Considering the increased ocean runup along our, particularly Maui seashores, should we, as people begin to consider cutting back on publicly funding flood insurance?
Well, I think people might need the flood insurance. Maybe we should stop building close to the shore. That would be wise. We've definitely been building way too close on the sand, but I'm not too sure about flood insurance. It'll be needed, I think.
We're paying for it.
Yes. But then a lot of the infrastructure we use, such as roads, lies along the beaches. So that is something we'll all have to pay for.
Another question. It's about coral. I was under the impression we had all this hullabaloo about the sunscreens, toxic, that it's the change in the water that's the problem, that's causing the majority of problems with the coral, not just the toxic materials in the water itself, but the fact that it's the temperature is going up. And so we can't do a whole lot about our sunlight. We're sort of, that's a given. So your particular, I can't remember the name of your coral, how does it fare in water that's semi-polluted and quite a bit warmer? Has it adapted to those situations?
Right, that's a good question. So that species is actually a very hardy species. So it does really well under conditions that would be very stressful for other corals. And that's because light and temperature are really correlated in the bleaching response. So it's actually the kind of mechanistic process of photosynthesis. You can either overheat the whole thing where all the proteins that keep it functioning break down. But if you bring the light down whilst that's happening, it can maintain itself at a healthy condition for longer. So one of the mitigation strategies for increased ocean temperatures is shading the ocean, basically, because if you bring the light down, they can tolerate higher temperatures. And so that's what we think is happening with this species, is that because it can reduce the light that it experiences, it can tolerate higher temperatures.
Just as a very brief anecdote, our lab went to Christmas Island, which is a couple of hours south of here by plane, and almost every coral there was dead, except for her coral.
Is that right?
Yeah.
Last question.
How would you guys handle, how you gather DNA? How would you guys know what species it is?
Well, so that's essentially where we have to know what a DNA sequence looks like for a given species first. So, there's a lot of that information is already out there. But if I didn't know, I would go out, right, and I would catch a fish, and I would say, okay, this fish is this species, right? So let's pick one. This guy, this Ulupiliku, right? And then I would clip a very small piece of his tail fin off, which doesn't hurt him. He can still swim. He would swim away. I'd take pictures, right? I would get the DNA out of that tail fin, and then I would put it in the computer, and I would say, okay, this fish goes to this DNA. And then you do that for basically everything that's out there as much as you possibly can. And then when you go back and you collect the DNA straight out of the seawater, you can refer back to that catalog that you've built. So there's a lot of stuff that we don't know what it is, right? But there's a lot of stuff that we do. So it's still a useful tool, and it's increasing all the time as we add more things to that catalog. Does that make sense?